Okay, so I have more important things I could be doing right now, so I'll get this done as fast as possible. No, I'm just kidding. This was lab number 5, the microscopic observation of cells by Soham and I. So first we'll talk about cells and what they are. Cells were discovered by Robert Hooke in 1665, and what he did is he used a simple microscope that he built himself to view thin slices of cork. And when he looked at this cork, he saw small compartments, which he decided to call cells, and the name stuck. The cell theory states that all living organisms are composed of one or more cells, that cells are the smallest functional units of life, and that cells arise from pre-existing cells. So more on the history of microscopes. The first microscope was a very simple compound microscope invented in 1590 by Zacharias Janssen. In 1893, Kohler developed a technique using illumination, a source of light, that served as, and this serves as the basis for modern light microscopy, so we still use it today. We used it in the lab. Anton van Leeuwenhoek is considered the father of microbiology because he improved the microscope and really made it mainstream in the scientific community. And Ernest Ruska, he worked on the first electron microscope in 1931, and I'll explain what electron microscope is shortly. So first, light microscopes. They're the most common microscope. They operate using visible light and lenses that magnify an object, and that's what we used in this lab. Then there are electron microscopes, which are fairly different. There's two types. There's the scanning electron microscope and the transmission electron microscope. The scanning electron microscope, it provides a view of an object using a fine electron beam, and the images can have a field of depth, which means that they can be three-dimensional. The image is three-dimensional, and the specimens viewed under a scanning electron microscope must have a metallic coating. The transmission electron microscope, it provides a view of an object by passing electrons through it, but the object must be a thin sheet, a very thin sheet, which means that the specimens viewed will always be dead. So then there are the parts of the microscope. This is a light microscope. There's the eyepiece, the body tube, the revolving nose piece with objective lenses, the stage, the condenser, the mirror, the base, the coarse adjustment knob, and the fine adjustment knob. The eyepiece, of course, you look through it to view your object, and the eyepiece usually, almost always, has a magnification in itself. In our case, it was 10. So then the revolving nose piece has 3 or 4 different lenses, in our case 4, that you can turn to magnify an object. Excuse me. In this case, for our microscopes, there were magnifications of the objective lenses of 4, 10, 40, and 100. And to find the total magnification, you multiply the magnification of the eyepiece and the objective lens. So we would have total magnifications of 40, 100, 400, and 1000. The stage is where you view your object or specimen, usually on a slide. The condenser is what allows light to pass through to the stage. The coarse adjustment knob adjusts the image by moving the stage. The coarse adjustment knob adjusts it much more than the fine adjustment knob. The coarse adjustment knob is used first because it, like I said, it moves the stage more, and you just get a good idea of how clear your image is. You just want to get in a good range, and then what you do is you use the fine adjustment knob, which, like it said, finely tunes the stage, moves it at very small amounts, so you can get the clearest image possible. There are some terms when using a microscope. There's the resolution, which is the ability to differentiate between two adjacent objects, or, well, two different objects. The contrast is the difference between two objects, or an object and the background. The field of view is the diameter of the circle one sees when looking through a microscope. And then there's the working space, which is the distance between the lens and the slide. So In this lab, we discussed biological taxonomy, which is the biological organization of organisms into different groups. These groups go from the least specific to the most specific, which starts with domain and goes to kingdom, phylum, class, order, family, genus, and then species. There are 3 domains. There's eukarya, bacteria, and archaea. And in this lab, we discussed the 6 different kingdoms, which are Animalia, Protista, Plantae, Fungi, Eubacteria, and Archaea, which I'll all discuss shortly. So I'm going to discuss prokaryotes and eukaryotes only because we did not really discuss Archaea in depth, and we didn't view any during this lab. So I'll compare these two. Prokaryotes, they're unicellular. They have a nucleoid region. They have a glycocalyx, flagella and pili, plasma membrane, a cell wall, and cytoplasm. Eukaryotes have a nucleus. They have membrane-bound organelles. They're compartmentalized, which means they're separated into different compartments. They have a plasma membrane. Some have cell walls, like plants, and some have vacuoles. So another example is plants. Here's a prokaryotic cell. As you can see on the inside, it has cytoplasm. It has a nucleoid region, which you can see greatly differs from the nucleus of a eukaryotic cell, which we'll see in the next slide, because this one is more spaced out and less dense. Here's the capsule, which is the glycocalyx, which is a gelatinous covering of the whole cell. And there's the cell wall and the plasma membrane. Prokaryotes also have ribosomes, like both prokaryotes and eukaryotes have ribosomes. They both synthesize proteins. Pili are small, relatively small projections of the cell, and they allow for movement. The same for the flagella, but there's usually only 1 or 2, and they're much larger, as you can see in the picture. Then there's a eukaryotic cell, which has a nucleus, as you can see, which is much more dense. And it has organelles like the smooth ER, the rough ER, flagellum. Only some animal cells have a flagellum, or some eukaryotic cells, I should say, have a flagellum. And this is an animal cell, just to be clear. This is just to differ from a eukaryote and a prokaryote. There are different kinds of eukaryotic cells, which we'll see. And other examples of organelles are the cytoskeleton, the mitochondrion, plasma membrane, Golgi apparatus, and ribosomes. Then there are 6 kingdoms, which fall right under domains. The kingdoms that have eukaryotic cells are Animalia, Plantae, Protista, and Fungi. The kingdoms that contain prokaryotic cells are Eubacteria and Archaea. So first we'll discuss Animalia, and then after this I'll get into what we observed in the lab that were animals, and I'll do the same for each kingdom and pretty much explain the procedure as we move along. So Animalia are eukaryotes. They're heterotrophs, which means that they feed indirectly or directly off of other living things, like how humans eat, I don't know, beef. Most animals undergo sexual reproduction involving meiosis. Some animals are also capable of asexual reproduction, which is parthenogenesis. An example of animals are humans. So here we looked at a human blood smear, which of course contain eukaryotic cells. Here's the blood smear at a 40 times magnification. This is the same blood smear at 100 times magnification. As you can see, the image is clear, but it's still very hard to see individual cells. So then we move to 400 times, and you can see here that the individual cells are much clearer. And also, I'm sorry, one more thing. In this, not only do we see red blood cells, which are erythrocytes, we see a couple white blood cells throughout the smear. Here you can only see, I believe, 1. But if you look under the when we looked under the microscope, there were more present. So next we had to prepare a wet mount to observe cheek cells. So when you prepare a wet mount, first you have, of course, your slide. Next we put a drop of methyl blue, which is a very strong dye. So then I myself swab the inside of my, well, I shouldn't say swab, used a toothpick to scrape the inside of my cheek so we could get cheek cells. And then we transferred those onto the methyl blue and carefully placed a cover slip over that so we could view the cells. So this is what it looked like at the 400 times magnification. Later in the conclusion we'll discuss why it looked like this. So the next kingdom is Plantae, and Plantae are made up of eukaryotic cells that are found everywhere. Plants are found everywhere. They are autotrophs, which means that they self-feed. They produce organic molecules from their surroundings. An example is photosynthesis, where light is converted into sugar. They could reproduce by either asexual means or sexual means. An example of a plant is the sunflower. And they impact human existence because humans eat many plants, like fruits and vegetables, of course. And plants intake CO2 and release oxygen as a waste, and that provides humans with oxygen. Of course, we need oxygen to survive. So the plant we looked at was Elodea, and here we see Elodea at 400 times magnification. As you can see, it differs greatly from the animal cells of the human blood smear, which again I'll discuss in the conclusion. The next kingdom was fungi. They are also eukaryotes, which can be found most places. They're organotrophs, which means that they obtain hydrogens and electrons from organic substrates, and that's how they power themselves, pretty much. Again, they can reproduce sexually or asexually. An example of a reproduction of fungi is by spores, and they impact human existence because some fungi serve as pathogens for humans, such as Aspergillus or Aspergillosis. And another benefiting way, is that humans have used penicillin, which is a fungi, to obtain the antibiotic penicillin, which we know has been greatly used for the past 60 or so years. So here's an example of a fungus. This is yeast at 1,000 times magnification. Here we saw Aspergillus, which is another type of fungus, at 400 times. Here you can see their heads. Here's I think a pretty good picture of Rhizopus, which again is another fungus, this time only at 100 times magnification. So the next kingdom I'll move on to is eubacteria. And eubacteria are made up of prokaryotic cells, and again, bacteria are found pretty much everywhere. They asexually reproduce. One way is binary fission. They are considered chemoautotrophs, which means they synthesize all necessary organic compounds for CO2, and they use inorganic energy sources. They impact human existence by creating symbiotic relationships with humans in the intestines, in which contribute to gut immunity and good digestion. But there's also pathogenic bacteria, which cause diseases such as tetanus, and there's many more. There's three shapes of bacteria. There's Spirillum, which is a twisted or spiral shape, Bacillus, which is rod shape, and Coccus, which is a spherical shape, which we'll see in the next few slides. And I will wrap up this first part of the presentation because my time's about to run out. So we will see the bacteria and the rest of the lab in the next video.